right, so in addition to lipids playing the roles that we previously talked about, basically making up membranes, lipids have major non-membrane functions. Those, or examples of those, are signaling lipids, lipid cofactors, and pigments or antioxidants. Okay, so signaling lipids are basically steroid hormones are a really good example of lipids that behave as chemical messengers. Lipid cofactors, if you go and take biochemistry 2, or if you go to medical school, or when you go to medical school and take a metabolism class, this is going to be of particular value to you. These are molecules that effectively are involved in the electron transport chain and oxidative phosphorylation. But if you don't know that terminology, you'll get to it soon enough. Likewise, lipids serve as pigment molecules. So as an example, vitamin A is an essential pigment molecule for vision. And one example of a lipid that you're probably very familiar with, but you haven't really thought about it in terms of classification, would be something like beta carotene. Beta carotene and basically any other major plant pigment molecule is an example of a lipid that has a non-membrane function. Now, first, what I'd like to get into are these eicosanoids. Eicosanoids are 20 carbon long fatty acids. They act at low concentration, so they're very, very powerful, and they're involved in the production of pain and fever, as well as regulation of blood pressure and blood coagulation and reproduction. Okay, so they're produced and used locally. An example of how they're broken down or how they're produced is the enzyme phospholipase A2, which we've already talked about. That's going to target that carbon-oxygen bond on carbon number 2 of a glycerophospholipid. That is going to release this 20 carbon long fatty acid. That 20 carbon long fatty acid is known as arachidonic acid. Now, what arachidonic acid does or undergoes is it undergoes this cyclization event right here. So this cyclization is catalyzed by enzymes known as COX enzymes or cyclooxygenase enzymes. Basically what we're doing is we're making this ring structure right here for the production of prostaglandins, this ring structure right here in the production of thromboxanes, and what we can also do is make this epoxy ring right here, forming the enzyme known as, the metabolite known as leukotriene A4. Now, what's significant about these is that these, since they are involved in, these molecules that I'm putting my stars by, these are involved in pain production and fever production, as well as regulation of blood pressure. So essentially, these enzymes right here, these COX enzymes, which I'm going to highlight that, these COX enzymes are critical for basically the production of fever. So what that means is you can use drugs like, non-steroid anti-inflammatory drugs, such as aspirin, to block these enzymes. So if you block that cyclooxygenase enzyme, then you block the production of prostaglandin E1, thromboxane E2, and therefore you're not going to develop any sort of fever. Okay. Now, ultimately, that has led to drug development, and Singulair and Zyflo are examples of enzymes that inhibit or prevent the production of leukotriene A4. But this sort of metabolism is something that's definitely very interesting and very significant for drug development. Okay, now cholesterol. Cholesterol is your basic molecule that's, or your basic, maybe the most popular lipid that does not serve a membrane function, or non-membrane function lipid. It's based on a core structure consisting of 3 6-membered rings and 1 5-membered ring. They're all fused together. Now, what cholesterols can have is a polar head group, and they can have different variations on that polar head group. We've got a steroid structure or steroid classification for this molecule, and what they're going to do is ultimately, cholesterol is the most common steroid in animals and precursor for all other steroids in animals. So if you ever do any sort of, like, one example is sex hormones are derivatives of steroids. If you ever look up the World Anti-Doping Agency's webpage, you can find a list of all banned substances, performance-enhancing drugs that athletes aren't supposed to be able to use, and many of them are based on steroids. Okay, so phytosterols are examples of plant steroids because plants do not have cholesterol. However, they do develop or they do synthesize sterols. Stigmasterol, beta cytosterol, and camphosterol are all examples of plant hormones and plant steroids. And so we can use them as basically regulators of cholesterol levels in humans. Okay, so your steroid hormones are probably the most well-known cholesterols. So, and generally because they have some, I would say, pop culture almost influence, because testosterone is something that you hear about if you watch any sports or anything like that. But steroid hormones bind very tightly to specific receptor proteins in the nucleus. Sorry about the typo there. They're going to regulate gene expression and metabolism. Major groups are the sex hormones, and those produced by the adrenal cortex. I would say that maybe the most popular or most well-known steroid hormones would be your testosterone and cortisol. Cortisol is something that athletes are going to take from time to time in order, they take cortisone. Cortisol is your stress hormone, testosterone is male sex hormone. And so these are all, like, derivatives of these are generally what athletes are going to use quite often, and any sort of modifications to these are going to be more than likely banned substances in any legitimate major sport.